Gonna wait a couple of more seconds and we'll start the session. Well, we're actually sharp 2 p.m. here in Lausanne. So hello, everyone. Welcome to this session of NeuroMatch conference. As I just mentioned, now it's 2 p.m. in Lausanne, but I hope you have a good time on every time zone you are around the world and you're safe. Well, you see that basically our session is going to be today in the format of traditional talks. So our speaker is going to present their work in about 12 minutes, and we have 3 minutes for questions. So please do ask your questions in the Q&A panel, and remember to upvote those questions since we might have many questions, and then we will at least have time to ask the most favorite questions. And you can also use the chat to share your ideas and thoughts and relevant papers. We have an amazing lineup of speakers in this session, and we're going to learn a lot about motor control from complex motor movements and flexible movements to effective behaviors. So I think we could maybe, with that, I would, let's say, make it short introduction and introduce to you our first speaker, Shreya Saxena. So Shreya did her actually undergrad studies here in Lausanne, where I'm based now at EPFL. So it's a double pleasure to host her in this session. And she recently started her own lab as an assistant professor at the University of Florida. And I think she will talk about neural strategies that produce flexible movements. Stage is yours, Shreya.
Thank you. Thank you so much, Vahid. And yes, we have that in common. We were both at EPFL at some point. You're still there. I'm jealous. I liked Lausanne a lot. And I'll get started today. So this is work that I did primarily during my postdoc at Columbia University. Thank you all for joining, and thank you to NeuroMatch for organizing this. This is a wonderful opportunity, especially in times such as these. So I'll talk today about network principles and how they can inform us about strategies in the motor cortex. So as a motivation, I want you to take the case where you're practicing a new dance move, let's say. So you first practice it slowly, get the steps right, and then once you have learned it, you can flexibly speed up the movement and make it at the speed that you would like. There are many other movements where this kind of strategy applies, and today I want to address the question of how the motor cortex may be achieving this goal. So to answer that question, a colleague, Abigail Russo at Columbia in Mark Hirschland's lab, trained 2 monkeys to cycle through a virtual environment while tracking a target. You see the target here on the screen. The target was continuously moving at different speeds, and in addition, depending on the color of the landscape, the monkey cycles forwards or backwards to produce the movement. You may be familiar with the cycling task from Abby's 2018 paper, but in that paper, in fact, the monkey was going at a constant speed, and this is in fact a different data set, one in which the monkey's speed changes continuously. So at the same time, Abby also recorded single unit neural activity from the motor cortex and intramuscular EMGs, muscle signals. Okay, so we first take a look at the muscle activity. This is the activity of one of the muscles during forward cycling. This is a rhythmic movement, so we chop it up into cycles, and this happens to be the slowest movement. And in fact, the fastest movement looks like this for this muscle. And as expected, the duration of the cycle is shorter, as you can see, but the pattern of activity changes as well. And similarly, we have all the other speeds in the middle. We bin the activity into 8 different speeds. And we also have the activity of many other muscles here. I'm just showing one of them, but there are many other muscles. And in fact, the biggest signals in the muscle activity, that is the top 3 principal components, look like this. And this, as you can see, is fairly tangled up. It's quite messy. But our question today is, how is this muscle activity generated? So we ask ourselves, how would a model generate this data? Or what would a network do? As I like to call it, WWND. So we used a recurrent neural network to simulate the activity with speed as an input. So this is the hypothesized input that would be coming into the motor cortex. And so now our network itself is a model for the motor cortex, since it has the same inputs and the same outputs as that brain region. Okay, so here I am showing you the activity of the trained networks nodes in PC space and principal component space for each speed. And this activity seems to follow quite a clear structure, which in fact looks very different from the EMG structure. This was an example network model, but we fit over 100 models, and I'm showing you 4 other ones here, and they all seem to have this structure. And there, in fact, seems to be extra signals generated in the network while producing the EMG. And in fact, these signals are important. And how do we know that? We can actually decode the neural signals better using the network activity than if we had just considered the EMG data. So here I'm showing you an example neurons firing rate in white, and the corresponding fit using the RNN PCs 1 through 3 in green. And you can see that the fit is quite good, much better than if we had used the EMG PCs 1 through 3. And it's not true just for this one neuron. In fact, single neurons as a whole are fit much better using the RNN activity as compared to the EMG activity. Okay, so I've hopefully convinced you that the RNN model is a good one. It can output EMG activity. That's what we think the motor cortex is doing. It can fit single neuron activity. However, I could have done both of those with a 10-layer deep neural network that would not have helped us gain any insight or any understanding of the computational mechanisms involved in generating this activity. So the last thing we need is, in fact, interpretability in our model. Why does it act the way it does? What characteristics of the model can we expect to be present in the data? Now, the computational mechanisms in the network are fairly straightforward to ascertain since we can simulate it. So for example, here I've visualized the flow fields and showing the 2D projection of those. And we see that if we give it this, here I'm giving it the slowest speed input. In fact, we have a locally stable limit cycle. So even if the trajectory has its initial conditions somewhere in the vicinity of the trajectory of the limit cycle, it traverses the smooth flow field and ends up at that limit cycle. What about the other inputs? So in fact, as we vary the amplitude of the input, we find ourselves in other limit cycles. So this is from the slowest to the fastest input and correspondingly the limit cycle. So the structure here seems to be an input-dependent limit cycle. So that's all well and good for the network, but what can we say about the actual data? So we need features of this activity that we can see in the data, as well as data-driven metrics to quantify the structure. Okay, so what are the features in the model that we should look for in the data? They all seem to have this feature of having big circles in the dominant dimensions and a separation between the trajectories. So does the data have these features? And if so, why? So to take you through that, I will first introduce a data-driven metric that we can actually test in the data. And that is a metric called trajectory tangling. This is a data-driven way to diagnose a system's robustness to noise and how autonomous a system is. And why are we looking at this metric? This is so that we can later actually calculate this directly given the data. So here, just to take you through what this is, take the state of a system, X at time t, and out of all of the other time points, check which has the largest difference in derivative, but is the closest in state. So here, that happens to be t prime. Basically, if it has a different derivative, it better be far away in state. Otherwise, it's easy for the trajectory to be knocked over to a completely different trajectory. So for example, here I'm showing a very tangled point. And this is the actual formula that we're using for this metric. And here are some trajectories with points of high tangling and some others with low tangling. So the important thing here is that this is a completely data-driven metric, as I had said before. And the goal is to characterize a system through only the observed trajectories. So what does this tell us about the motor cortex and the muscles? We believe that the motor cortex has noise-robust dynamics, and thus that the motor cortex has low tangling. And in fact, the rhythmic trajectory with the lowest tangling is a circle. So that's our first prediction. So here I'm showing you the network activity again, and this gives us a concrete reasoning for the first geometric feature of big circles in the dominant dimensions. Now, if these were the same big circle in high D space, we would have a tangling problem because we would have trajectories which have different derivatives but are very close in state. So we don't think that's happening either. And that leads us to our second geometric feature, that these circles are well separated in high dimensional space. So this gives us two concrete predictions for the data. So let's see if these exist in the data. So to check our first prediction, we take the monkey cycling forwards at different speeds. And as a comparison point, I'm plotting the muscle trajectory here. So each plot here is for the trajectory at one speed in its own PCs. And we see that the muscle activity is changing its pattern very drastically across speeds and has a lot of points of high tangling. Now to compare, we come to the neural activity. And here, no matter the speed, we in fact see a big rotating circle in our data. So that basically shows us that the first prediction that we had made is true in this data. And here, just to see how similar these circles are to one another, just overlaying them all for you. But remember, these are all in their own PCs. So I showed you that the neural data contains big circles in dominant dimensions that contribute to having low tangling per speed. But let me remind you, if these are the same big circles overlaid on top of each other, we would have a tangling problem since these would be going at different speeds or different derivatives. So we expect the separation between the trajectories. So let's just check. So here I'm showing you the different trajectories now in a global PC space. The first two PCs follow the same big circular trajectories as the network. But as soon as we get to the third PC, this is when we get to the separation between the trajectories, which is a large one. And we see that in the third PC directly, but we can also check that it does exist in the full neural dimensional space. And here we can directly calculate that. I'm just displaying the Euclidean distance in all dimensions from a reference trajectory as a function of the phase of the cycle, theta. And in fact, for every theta, the distance is monotonically increasing as a function of speed. So there is that separation between these trajectories in the neural space. But why is that important? In fact, I'm overlaying this now with the value for tangling in the global PC space, and you can see that the tangling remains very low for neural activity as opposed to the muscle activity here. There are many points of high tangling. And how do we know that it's the separation that is leading to low tangling? We can actually test that out directly. So here here's the within speed tangling for both muscle and neural data, and here's the 99th percentile of just the neural tangling values. And I'll first just show you how bad the tangling could have been if the trajectories were just overlaid on top of each other. Here we just speed up and slow down the reference trajectory and add biophysical noise, and they are basically directly on top of each other. And we see that the tangling in these simulated trajectories shoots up more than 2x. And in fact, the actual global tangling is nowhere near this level. And in fact, it is exactly this separation that is leading to the low trajectory tangling, which we can directly test by artificially removing this separation, in which case the tangling shoots up again. Okay, so with these data manipulation techniques, we can see that there is indeed a large separation between the trajectories at different speeds, which is exactly the thing that is leading to low tangling across speeds.
Shreya, sorry for interruption. If you could wrap up, would be great. We have time for questions. Thanks.
Sounds good. And so I am basically at the end with these two main geometric features, both the network data and the neural data make sense. And this all came from what would a network do at WWND. So with that, I want to acknowledge my co-authors and thank them, Abigail Russo, John Cunningham, and Mark Churchland. And look out for the paper that is forthcoming and soon on bioRxiv. Great, and if you're interested in questions such as these, check out my lab. I have a new lab, as Vahid mentioned. I'm looking for collaborators and PhD students, and would love to just talk to anyone who would like to learn more about either this project or any of the other projects that I'm working on. And thank you all for listening.
Thank you very much, Shreya, for the great talk. I hope we have time for we have a few minutes for a couple of questions, so I'm going to go quickly to the questions. So our first question is from Barbara Fulner, and Barbara is asking if could there be missing dimensions from the muscle, like which you did not record from, which makes the EMG trajectories less tangled?
Yeah. We did record from a large number of muscles in the upper limb, around 30 or so. So we hope to have captured a lot of the variations in the muscle activity. That being said, yes, of course, that's always a possibility. And we did basically, we downsampled, let's say, the number of neurons, and we kind of made these kinds of, we did tests to check things like that, to check that the relationship between the neural tangling and the muscle tangling can be appropriately done. So that's something that we did take into account. But thank you.
Thank you for the
I
answer.
see that I answered another question from him as well. So.
Yes, exactly. So I was about to actually ask more of maybe a naive question. I think you showed us a beautiful metrics and showing this robustness to the noise that could be the reason of this low tangling in this dominant dimensional space, let's say dynamical space. But could there be anything also related to the nature of the movement, which is also a cycling movement that could have you tried that in like any non-cycling movement and see if you could find some, again, circles as the dimensions of the neural activity?
Yes, exactly. Now that's a great question. And I actually have a backup slide for that, which I'm trying to pull up very quickly. I understand that we're short on time, but just humor me. So your question is basically, is this motor cortex activity just encoding dynamics? But this is where that the kind of nuance of the experiment, where the monkey was doing both forward cycling and backward cycling, that kind of, so this was more explored in other papers, but it's definitely about, definitely a great question. So basically the idea is that the neural activity during both forward and backward cycling is actually just going in the same direction, is co-rotating. And that kind of convinces us that this is not just encoding for kinematics, because if it was, then these would be oppositely rotating or rotating in different directions. And that's
great answer.
That's the short answer. It also has been shown, rotations have been shown for other kinds of movements as well. So to answer your direct question, yes.
Thank you very much, Shreya. Thank you for the great talk and the questions and the audience. So for the interest of time, we have to move to our next speaker. I thank again, Shreya, on behalf of all the attendees. So our next speaker is Joe Lushinger. He is an MD PhD student in the lab of Danny Wender at Vanderbilt University. And Joe will talk about a novel pathway that regulates affective behavior. The stage is yours, Joe.
All right, thanks so much. Yes, so let me share my screen. There we go. And all right, can everybody? I think you should be able to see that now. So yeah, today I'll be talking about a pathway that runs from the insula to the BNST, its role in stress coping, and then how we identified a novel motor input to that affective pathway. So our lab is at Vanderbilt University, and we're focused primarily on addiction and how stress can interplay with addiction. We know that stress is a key component for inducing relapse in those who are trying to remain abstinent. And so we're trying to understand how that relationship works. I think everyone around the world and along with us in the United States have known that this has been a particularly stressful year for a number of reasons, many of which are impacting everyone. And I think especially us in the US have some apprehension about what is coming forward in the next couple of weeks with respect to the election. And we've seen a concomitant increase in overdose deaths. And so we know this is a huge problem. And so understanding how these are related is really, really critical. Now, the work that I'll show today is looking at 3 brain regions, starting with the bed nucleus of the stria terminalis, shown here in blue. It's primarily thought to process anxiety and sort of diffuse stress-like states. The insula, which processes higher order sensory processing and interoception, and then the motor cortex, which obviously helps drive motor behavior. The pathway between the insula and the BNST, we've shown some work on that previously, and so we're sort of picking up on that work, which showed it to be involved in negative affect. Specifically here, we're focusing on coping behavior, and this is really important because it's really critical to every animal's ability to adapt and survive, the response to stress is. And so finding an appropriate stress response shifts metabolic resources towards the critical systems that are involved. And so for our work, we're looking at mice. And so you can imagine a stress response that a mouse in a field, for example, in a predation scenario where they are experiencing the threat of a predator. They have a couple of options. One, they could freeze, which saves them energy, or they could flee, which is more of an active escape behavior. Fleeing removes the stressor, removes them from the stressor, whereas the freezing helps them avoid detection. And even though you get the same stimulus, you can have opposite, effectively coping strategies. And so we think this is an important branch point. So to try to understand how what signaling is happening during this change, we devised this restraint device that allows us to record in vivo fibrophotometry recordings. So this is fluorescent sensor-based recordings that are in the animal while they are undergoing this restraint stress. And so this allows us to measure their neural activity over time, essentially. Now, the way that works is we can inject a virus into a brain region of interest that codes for a fluorescent sensor that, when bound to its ligand, will emit emit light. So we can put the fiber optic cable down into that region as well, and we can fire an excitation light through a fiber optic cable down into the mouse, which is in the tube. And when the ligand, calcium, for example, binds to one of these sensors, it will then emit light, and we can capture that via this fibrophotometry system. We can also then time lock that to video that we've recorded of the behavior. And so we then use machine learning based tracking. Some of you may have heard of deep lab cut. So we use that to track different parts of the animals and then identify coping bouts. And so those coping bouts, what they look like when we are recording them, you can see here we're tracking both the head and the tail. So the head is in the dark blue line, the tail is the dark red line, speed is on the y-axis, and x is time. And what you can see is there are periods of sort of quiescence, and then there are large periods of fast movement. And so then we can just threshold and make those movements or bin those movements into different types of coping bouts: head only, tail only, or full body. And in this case, we're looking specifically at full body movements. The first area that we looked at was as I said, the bed nucleus of the stria terminalis, and specifically stress responsive cells called corticotropin releasing factor cells in the BNST. And I think if you look at these two, we've got GFP controls on the left. On the right, you can tell very different for orientation. In the top graph, you can see that there is essentially 0 is time locked to when the bout movement starts, and then you have going across each individual bout. And what's fairly apparent is you have an increased Z-score when you have with the onset of those bouts in the CRF cells, and they got a calcium sensor in them, and so that's a correlate of neural activity. And if you look down at the bottom graph on the right, you can see the average trace across those is significantly different than our GFP controls. So the question then that we had is, how are these stress responsive cells recruited? And we hypothesized perhaps there was some glutamatergic input that was driving this, and so we used a glutamate sensor called GluSniffer, and found a very similar pattern in that we have an increased glutamate signal in the BNST during these bouts. And so the next question was, where might these be coming from? We did some work that I won't show here, just to identify the insula as one of those candidate regions, and then to identify specifically where in the insula the densest input to the BNST was. These are just statistically showing that there was significant difference between those and the controls. So once we identified that really dense input from the insula, we then injected sensors into the insula, starting with a calcium sensor called GCAMP to look at what the neural dynamics were during these coping bouts. And again, when we time locked to the onset of those bouts, we found this very reproducible pattern in that we get this slight dip before the onset of behavior, and then a large increase that happens with that is time locked to behavior. And this is just general calcium signaling in the insular cortex. And so we're wondering what might be responsible, what other signaling might be responsible for this change that we're seeing. And so we looked at GABA signaling via a GABA sensor. So this is a sensor that senses extracellular GABA, and saw a very different pattern, which was pretty exciting in that we see a decrease of GABA signaling associated with these movement bouts. And so what we think might be happening is perhaps that dip is the GABA neuron sort of shutting down before the movement onset, and then the dip in the yellow line is sort of the washout of the extracellular GABA, and then you have an increase in the global insular signaling and then some of those projection neurons could be heading to the BNST. The next component, we did some other behavioral work to show that that circuit could modulate affective behavior. And so we were interested in what was the control network for this pathway. And one way to do that would be to hypothesize regions and then slice the brains up and look to see if we could find connectivity pattern. But we felt like that was sort of looking under the lamppost for our keys because that's where the light is. And so we wanted to use a more unbiased approach. And so we collaborated with the Simmerly lab at Vanderbilt to do some light sheet microscopy. And for those who are not familiar, the way light sheet microscopy works is you take a brain that you've fixed and you clear it by pulling out the lipids. You can then put it into a refractive index matching solution so that light can pass through undisturbed. You can then pass a thin sheet of light through the brain and image planes of those brains and stitch those planes together into a three-dimensional reconstruction of that brain. We can then put those 3D reconstructions into a uniform atlas and quantify where the cell bodies are. And so this gives us whole brain analysis of the inputs to the cells of interest. And so specifically what we used was a rabies-based tracing approach where we injected rabies into the insula. And we did a couple of other viruses that I can go through in the questions. Essentially what we're doing is we are restricting the entry of rabies into the cells that project to the BNST from the insula, and then restricting their ability to jump backwards one synapse from those cells essentially only one time. And so it allows us to get the upstream control network globally across the brain that is a single synapse away from this particular pathway. We can then do that across a number of brains. This is the sort of top hits. This is actually a very long list of tens of brain regions, but we'll notice there somatosensory is the top hit there in both the raw cell count and then normalized to cells per brain on the right. Not surprisingly, since it does so much sensory processing, but one that was surprising to us a little bit was the somatomotor areas and how in both cases they were some of our top hits. And so we're interested in that because of exercise and movement's role in one, avoiding a stressor, and two, exercise role in alleviating stress and the antidepressant effects of exercise. And so once we found that, we were pretty interested in this pathway, and so we wanted to verify that this wasn't some type of spurious labeling. And so we used another convergent technique to virally label from motor cortex the outputs and the inputs to the BNST, with the idea that the overlapping cells would be doubly labeled. And so I'll just show a brief video here, which is showing this is a light sheet brain, so we're peeling away the surface here so we can look down inside the brain. These are the two injection sites. The cyan cells are the cells that are labeled with that are getting inputs from motor cortex, and then the magenta are getting inputs from motor cortex, and the cyan are projecting to the BNST. So we're looking for white cells or overlapping double cells. And I think what you'll appreciate here, we're now taking a plane through the brain that's 150 microns, and you'll see some double labeled cells that are in white, indicating, yes, we do in fact have overlapping cells that are in the insula that go to the BNST and receive motor input. And so I'll just move on for the sake of time. You can see there were a ton of cells there, and so we wanted to be able to show that they were connected, but patching them was going to be difficult. And so we devised another strategy to retrogradely label only cells that get motor input and go to the BNST and nothing else. And the major takeaway here is
Joe, sorry, just for the interruption, if you could wrap up would be great. Thanks.
Yes, great. So we got this shows that there is structural connectivity, and then we also went on to show that they were functionally connected with electrophysiology, and the vast majority of them were. With that, I'll just thank everyone that was involved. The folks that are underlined here played a really key contributing role. And with that, I will take questions.
Thank you very much, Joe, for the very beautiful talk and extensive work. So while I'm waiting for the questions to appear in the question and answer, I have the first actually thought. I was looking at this pattern when you look at the inhibition, like when you look at the GABAergic responses. Mm-hmm. And I'm again quite naive in this field, but how much do we know? Like, because we hear now a lot about the acetylcholine and the effect of modulation of acetylcholine during arousal, and would that also have an influence in this case, like a disinhibitory effect? Again, I don't know how many of inhibitory cell types do we have, where you're looking at, and would it be somehow that there's again VIP neurons somehow inhibit the somatostatin neuron and they would then disinhibit? So somehow would that go in that direction, do you think?
Yes, so I can tell you this is fairly newish data, and so we've not explored much beyond that. But your question's a good one, which is what's driving that change in GABAergic signaling, and so that's something we're looking to explore sort of next, is to figure out what population. So we've done some preliminary tracing work from motor to look at what those might be, and it looks like maybe somatostatin. Now, we haven't looked sort of extensively at all the different types of GABAergic neurons in the insula, but certainly some of the somatostatin neurons in the insula are getting motor input. So we're thinking perhaps there's sort of some feed-forward inhibition going on that could be sort of taking the brakes off of some of that as well. As for the acetylcholine signaling, that's a good question. I haven't actually thought about that too much. And so I don't have a good answer for you, but it's something I'm going to do some reading about.
Thank you for that.
I have another question,
yes,
maybe on the same note, but again, a very naive question, but the connections from the somatomotor, would they make sense based on the stress response, due to the stress response? Because if the animal is already fleeing, let's say, this area should know that, or is it very
No, that's a great question. So we've given that some thought as well, and the short answer is the next component of our work is going to try to, I think, look more at what the function of that motor input is. One of the things we think about is sort of motor efference copy, and so one to playing the obviously motor cortex is driving what's happening in the body, but sort of the same reason you can't tickle yourself. Perhaps during this fight or flight response, the motor cortex would also be sending information to your interoceptive system to tell your body that some of the changes that you're sensing are because we're actually engaged in some motor response.
That's exactly what I was thinking.
Yeah. So do you have thoughts on that? I'd love your input.
Yeah, I mean, I think that sounds like a very reasonable way to do things for these different brain regions. So yeah, I'd love to know more as to. I think the timescales matter, right? So you were talking about the effect of exercise. That's at a much longer time scale than these kinds of inputs that you might be getting at a shorter time scale, just based on what's happening at that moment. So that would be super interesting to find out.
Yeah.
I'm very curious as to the results. Thank you.
Yeah, we're planning to start doing some work with longer active bouts. So add more of an exercise-like paradigm to look and see how that either has similar or different signaling patterns.
Great.
All right, thank you very much, Joe, for the great talk and answering all the questions. Again, for the interest of time, we're gonna move to our next speaker. So next up is Chloé Delepine, that she will talk about the contribution of astrocytes to motor learning. And Chloé is a postdoc in the lab of Mriganka Sur at MIT. Stage is yours, Chloé.
Thank you. Yeah, it's been an amazing session so far, and I'm gonna finish it with yet another perspective on the motor cortex, and this time focusing on the astrocytes' contribution to motor learning. Oops. Okay. And so astrocytes, as you may probably know already, are cells that are tiling the brain and that contributes to brain function via multiple ways. That has been evidence for their role in metabolism, blood-brain barrier, synaptogenesis, immune response. But what I'm particularly interested in is to study their role as responding and regulating neurotransmission, notably through neurotransmitter clearance. And there is evidence also that astrocyte responds to neuromodulators via expression of G protein coupled receptors and calcium signal. And all of this, there is evidence for all of these functions, but however, there is only a few in vivo studies that have been focusing on the astrocyte neuron interaction during behavioral task and learning. And so that's what we decided to focus on for this postdoctoral project. And I've been decided to choose motor learning as one of those complex behavior to focus on. And so motor learning, in my definition of it for this project, is the acquisition of a reliable and dexterous movement. And it's been shown that the M1 cortex, as you've seen before, is involved in motor learning, including the acquisition of the task and also the performance of the movement. Motor learning is associated, at least in rodent, with the emergence of a new ensemble of neurons, which are a subset of highly correlated neurons that are active during the learn movement. And we decided to use this quite simple lever push task that was described by the Komiyama lab first, where a mouse is receiving a reward when it pushes a lever to a certain threshold after a cue tone. And we've been asking the following questions. So first, we know that neurons are functionally remodeled during motor learning. What about astrocytes? Are they also changing during motor learning? Can astrocyte modulate motor learning? And can astrocyte modulate the neuronal changes associated with motor learning? And to answer those questions, we apply various strategies. Okay, so first, are astrocytes changing during motor learning? To answer that question, I've purified astrocytes from M1 cortices of mice that have been trained to different stages of the learning, so either naive, novice, or expert mice. And I've discovered that around 100 gene sets are indeed changing their expression during motor learning. And here is the most dysregulated gene sets that you can see. And what you can notice is that most of them are sets of genes that are transporters, transmembrane transporters, and most of them are overlapping by including glutamate transporter genes, such as GLT1 and GLAST. So that's the first thing. Then Jennifer, with also a postdoc in the SIR lab, has been looking at calcium signaling in astrocyte microdomain using in vivo chronic imaging of mice trained to this same lever push task. And our main result is to show that in an expert mouse compared to a novice mouse, you see an increased coincidence of calcium events in the microdomain during the push, during the lever push. So we've been showing that yes, astrocytes are changing during learning. They show some kind of plasticity that include probably other things, but we identify that it's including glutamate transporter gene expression changes and astrocyte calcium activity changes. So then are astrocytes able to modulate directly motor learning? To answer that part, we decided to target astrocytes using genetic and chemogenetic manipulation of their functions. And we use two different models. The first one is to knock out the glutamate transporter GLT1. So it's the main glutamate transporter expressed in the cortex and mostly expressed by astrocytes. The other tool we used is to use an engineered GQ-GPCR receptor that can be activated by an exogenous ligand, CNO. And so this was used by, using flux GLT1 mice with local injection of the Cre directly in the motor cortex. And here we directly inject the viral expression of the GQ dread. So what are the effects of those manipulation on behavior? First, for the GLT1 knockout mice, we showed that actually the mice are still able to learn the task normally. So this is their hit rate, this is their response time. It's similar to wild type. However, if you look at the lever push trajectory that are shown here, you can see that the GLT1 mice do not learn a dexterous movement. Their movement is not stereotyped nor reliable. And the GQ dread animal, the GQ activated animals, on the contrary, and so if it was not clear, this GQ is specifically expressed in astrocytes, of course. So in those animals, we see actually that both the performance and the movement dexterity are impaired. So slightly different result than the previous model. And in this model, we were able to try to inject this controlled saline solution instead of the activating CNO. And we see that in that case, actually, you can rescue the performance. So yes, astrocytes seems to be able to modulate motor learning, and we showed that knockdown of GLT1, so glutamate clearance, decrease of glutamate clearance will trigger an impaired dexterity, but will not impact the timing, the response time of the animal. However, GQ activation of astrocytes impair both the dexterity and the timing. So then we focused on neuronal activity and with the same two models, assess what were the effects on neuronal activity. For that, we used in vivo chronic 2-photon imaging again, but this one of M1 neurons, and by using GKM6S calcium sensors. So with this technique, you can image hundreds of neurons simultaneously during the performance of the task. And so we looked at that in expert animals. And so from what we can see with looking at calcium sensor fluorescence, is not much difference in the activity itself, but what we could see is a decrease of the interneuronal signal correlation during the movement period. So that's what you can see here. More neurons are with low interneuronal correlation compared to in wild type, where you have at least a group of neurons that are highly correlated. In the GQ activated mice, again, not much change for the activity, but we see, on the contrary to the GLT1 mice, we see an increase of the interneuronal correlation. As you can see here, there is a shift for more neurons being highly correlated. So then we wonder if we could look at how the motor cortex neurons are encoding different task parameters during this task, and if we could see differences in the encoding of the task parameter looking at our two models of astrocyte modulation. And this was work done in collaboration with KG Lee, who's been running all those modeling. And one thing that we've been looking at is using GLM models and measure their performance to predict single neuron activity from those different task parameter. So that include trial event, like the start of the trial, the reward time, also the lever movement, so the trajectory, the speed, and finally, if the trial was a success, and the response time. So when you take all those predictors together and you look at the performance of the model, you observe that both the GLT1 and the GQ animal have a lower performance of the GLM to predict the neuronal activity. But if you look at the contribution of each of those variables, first to the performance of the model, you can see first that in wild type, we identified that the parameters that are the most encoded or the most contributing to the neuronal activity encoding, are the success of the trial, the response time, and the speed of the movement. So that was the first interesting result. And we see actually that in the GQ animals, it's mostly the same contribution. The distribution of the contribution of the variable is mostly the same. But if you look at the GLT1 animals, it seems that the response time is contributing proportionally more than the other variables. So that was interesting. And actually, so if you remember the behavioral result that I show you, this could be a nice representation, or nicely associated at least, with the fact that the timing in those mice, the response time was not impaired. Only the dexterity of the movement was impaired though. So that's interesting. So yes, we can see that when you modulate astrocyte functions, and notably the glutamate clearance and the GQ signaling, you do see a change in neuronal correlations, and you see a decrease of the neuronal encoding of task parameters, notably the response time and the movement speed. So in conclusion, M1 astrocytes do show in vivo plasticity during learning of a lever push task. And we've shown that by transcriptomic and functional analysis, astrocyte manipulation of M1 are sufficient to alter motor learning and execution of the movement, as well as the neuronal population coding, again in the lever push task. And finally, we demonstrate that astrocytes have a specific role in coordinating M1 neuronal activity during motor learning, and to control the learned movement execution and dexterity through a mechanism that seems to include fine regulation of glutamate transport. And with this, I want to thank you for your attention, and all the members of the SOAR Lab, my collaborators, and financial support. And I'm happy to
Thank you very much, Chloe, for also being exactly to the time, perfect timing, and great talk. So while I'm again waiting for the questions, I'm gonna actually ask you to a little bit elaborate on the interpretation of so the manipulations you performed that you showed us that are partly different results depending on the, like in the GLT1 mice or the GQ mice, I think, that when you use DREAD, or and then also the correlations that are like, one is decreasing the correlations in the neural activity, one is increasing. Like, how do you put this together, and then the impact that they have on behavior, on the motor behavior?
Yeah, the parallel between the two is very interesting, and we are still trying to figure that out. But so yeah, it seems that the two models are opposite in some ways. So GLT1 being decreasing the correlation, GQ DREAD activation seems to be increasing the neuronal correlation. But in other way are the same. Like in both animals, the animals are not learning well to do a reliable movement. So there are some overlap and some opposition. So we are trying to look at that by, so for example, we started observing that the GQ DREAD animal also show actually an increase of the expression of the GLT1 gene. So there could be some molecular mechanisms that are indeed opposite in the two models. But also my global idea of this is that if you perturb the motor cortex you will, so the correlation of neuron in the motor cortex is the very important part of the motor cortex function. And so if you dysregulate it, if it's too correlated or too less correlated, in both cases, you will prevent motor learning in some ways. And then it seems that the response time is affected differently. And for this, we don't have any mechanism to suggest yet, but that's very interesting that in one case, the response time is preserved and not in the other case.
Thank you very much, Chloe. Well, I have one more question. I think we have quickly, if you could shortly answer this question. So the question is from Gregory Handy, and great work, Chloe. And what are your future steps in investigating these pathways? For example, can you do GLT1 knockout and GQ DREAD at the same time?
Mm-hmm. Yeah, it's a question that I've been asked several times now, and it's definitely in my plan. Actually, I've started gathering some mice to do this. It's gonna be my next experiment. Yeah, thanks.
Okay, thanks for the great question and great answer. And with that, I would like to again thank all the speakers, Sharia, Joe, and Chloe for their great talks. And again, another round of applause for, again, on behalf of all the attendees, for all our speakers. Thank you very much, also the attendees, for your questions and I hope you enjoyed this session and you would enjoy the rest of the conference. I would like to also thank Saeed for that let us run this session quite smoothly with his technical support. Thank you very much, everybody.
Thank you.
As the people are joining in. Okay. Should I start, Fazula? Okay. Okay. Welcome everyone to this session. I'm Gaijin, I'm the MC for this session. We have 3 amazing speakers in this session. They're going to tell us about how the brain segments information into structured event representation, about synaptic plasticity over distinct time scales, and about Hubble plasticity memory consolidation. It seems they are very related with each other. I'm not working on it, but it's so fascinating. So very much looking forward to their talks. And I'm sure we are going to learn a lot from them. Okay, our first speaker is Omri Radka. Omri is a PhD candidate from NYU, working with Professor David Purple. He does a lot of cool stuff on auditory sequence representation, memory and learning, large-scale brain networks, and intracranial EEG. He had publications out on Nature Human Behavior, Nature Communications, etc. If you need to reach him out after the talk, it's very easy to find him on the website. I did that this morning too. You also can find him on Twitter. Okay, please do remember to click Q&A to ask questions, and you also can upvote questions you like. You're also welcome to ask questions on the screen. Looking forward to your talk, Omri. Please take the stage.
Thank you so much for the introduction. Let me just share my screen here.
Sure.
Can you guys see my screen okay? I know it's a little small, but we'll be able to zoom into these boxes here.
Yeah.
Okay, great. Let me just do that. I'm teaching now, so I have all these extra Zoom skills. Okay. So, okay, let me just get started, although I think people are still funneling in a little bit. So thank you for having me, and thank you for the host. This is really exciting, and I'm really happy to be here. I'll jump right in because we only have 12 minutes before questions. So just some background. You know, we experience our perceptual environment seemingly continuously, and the memory system must somehow solve the problem of segmenting this information into composite events in memory. This is a function that we call often event segmentation. Okay, previous work has shown that contextual stability, whether in the goal state or in perceptual features of the stimulus, serve to temporally bind, so over time, bind items into composite events in memory. And this is primarily studied in visual paradigms and spatial memory paradigms. But in the current study, we wanted to take this into the auditory modality and study the role of both perceptual and semantic context for event segmentation for auditory information. We think this is important because an account of event segmentation, in addition, will lend to elucidating aspects of sequence memory and representation for complex auditory signals like music and language, and also teach us some general things about event segmentation. Okay, so how did we plan to study this? So we have a stimulus set of about 500 unique nouns, or common nouns, which are equally grouped across our blocks by number of syllables and written frequency. One thing that's really nice about the stimulus set by Gwyneth Lewis, who was a previous grad student in our lab, was that you can group these stimuli also into semantic categories. There's 66 semantic categories in the stimulus set, like mammals, tools, and professions. And what I had done is I had recorded these stimuli with 4 speakers, 4 lucky research assistants in our lab. So 2 female speakers and 2 male speakers. Okay. And we did 2 experiments with these stimuli. In the first experiment, we tested perceptual boundaries. So here you see an example of 9 trials out of 24 that exist in a block. And you can see that each set of 6 words is said by a different speaker. Okay, so that's how we're creating perceptual events or acoustically bound events. And we're always going male, female, male, female. Hope this makes sense to everybody so far. And the memory tests that we probe here are canonical memory tests for probing event segmentation in the visual modality. So we have within order memory. So we asked, did tennis come before soccer? And across order memory, did missile come before copper? So you can see they're spaced by the same amount of items and over time, but one is within a perceptually bound event and one is across. And our prediction is that within memory will be better if you're doing some sort of segmentation process. In addition, we have a source memory test. Was this word said by a male or female speaker? And that's either for the boundary item and the transition from one event to another, or the non-boundary item. And again, from previous literature, we anticipate that the boundary item, if event segmentation is truly occurring, will be better remembered than the non-boundary item. Okay. And then in a second experiment, we use the same stimulus set, but we shuffle the speakers. So there's no consistent speaker events. And each set of 6 words belongs to a different semantic category. So you can see here, there's an animal category, dog, horse, bear, cat, and so forth. And then it transitions to a vehicle category, van, jet, truck, and so forth. And so you have 24 items. That's 4 semantically bound events with the speakers shuffled. Okay, and then we do the exact same memory tests. So let me focus now on the results just from experiment 1. And then one other thing to note is that this is a completely passive listening task. So most of these experiments in the other modality mostly are concerned with, they ask participants to come up with a strategy, and they have participants also do an attentional task throughout. This is completely passive listening. Okay, so the first thing to note is that we're able to directly replicate the behavioral effects in the visual modality of event segmentation. So we see better within memory for temporal order than across, and better source memory for the boundary item than the non-boundary item. Okay, this is a direct replication. This was not obvious, right? We are completely switching modalities. And in addition, where this is completely passive listening, which hasn't been done before. And so we're able to replicate the results. If you're interested in seeing these figures in the visual modality, see Andy Hauser's paper from Leo Devacci's lab, which I cite here. Okay, so we replicate the findings, and now we want to go one step farther. So because this is a passive listening task, it allows us to probe what sort of mnemonic strategies are naturally adopted by participants. The way we did this is we do a post-task questionnaire where participants are able to rank on a 1 through 5 scale which strategy they use to remember the order of the words. Okay, so one strategy that's commonly used in the visual modality is associative binding. So they ask participants, or they suggest the strategy, I imagine the words interacting with each other to memorize their order. Okay, another strategy is repetition. I continuously repeated the words in my mind to memorize their order. We added storytelling. I created stories to memorize the order of the words. And finally, here we have a sort of proxy for the method of loci. I use imagined spatial cues and landmarks to memorize the order of the words. This was added in later because participants were telling me, you know, I'm putting objects in different places in the room. Okay, so let's see how these strategies affect memory performance in the task. So what happens if we take all order memory? So we take within and across order memory. What we find is that storytelling and method of loci best predict whether a participant will perform well in the order memory task. Interestingly, we see an inverse relationship with rehearsal and a weaker correlation with associative binding, which is often employed in prior experiments. Okay, so order memory is selectively modulated by these strategies, but what about segmentation itself? Our measure for segmentation really is how much within is modulated relative to across. So what if I plot within minus across as a function of these different adopted strategies? We basically get completely flat lines. Okay, this was quite surprising to me. And what this indicated to me is that the strategies modulate quite significantly the order memory performance, but seem to have no effect on actual segmentation. Okay, which means that the segmentation is not driven by the endogenous memory mnemonic process that's going on in the subject's head, but rather only, sorry, it's not driven by that, but it's rather only driven by the stimulus itself. So no matter what's going on in the head doesn't really seem to matter. Okay, and source memory shows very similar results. You can take my word that if we do boundary minus non-boundary, you again get completely flat lines. So the segmentation doesn't seem to be affected. All right, So now I'll move to experiment 2. This is the semantically bound events with the speakers shuffled. The first thing to note is that we are unable to get these canonical segmentation effects. So we don't see greater within order than across order. Okay, so we began to think, what's going on? Is it that semantic boundaries don't cause events in memory, right? We did not want to concede this quite yet. And so one potential possibility was that some of our categories are more coherent than other categories. Okay, so we have a category like toys that has the word sandbox in it. Okay, I'm not sure that I would consider sandbox a good category member for toys. Okay, so what we did is we employed a semantic similarity decoding approach here, which I'll show. And so for those unfamiliar with semantic embeddings, I'll just give a brief overview, and I'm happy to answer questions about it later. But basically, we're using Word2Vec here, which is developed at Google. And what it does is it scans hundreds of thousands Google News articles. And what it returns to you based on co-occurrences of words is a really high dimensional representation of a proxy for the word semantics. You can take this high dimensional representation, project it in a 2-dimensional space, and understand and get a measure for semantic similarity between words. Okay, if that seemed like a lot, what you have to take out of this is that this method allows us to get a proxy for semantic similarity between 2 words. How semantically similar are they? So here you can see our apparel category, which has jeans, belt, boots, shorts, dress, and so forth. And we plot it against itself, and we get the semantic similarity between each of the words in the category and take the mean of this matrix for how semantically consistent or coherent a particular category in our task is. Okay, and I do this across all 66 categories in our task. You can see we get quite a wide distribution of semantic similarity for our different categories. So what happens when we actually plot this against order memory performance? We see this robust correlation. So the more semantically coherent a category is, so we have some categories that are very strong, like animals and apparel, the better order memory is within an event. So this made us think, okay, well, look, maybe for some categories that are strong, where you transition from, where you also have strong transitions, you do have this event segmentation effect. Okay, so can we actually go more granular than this and observe whether this is happening? This is what we do here. So our approach is as follows. We have 9 trials I'm showing you here. There's 24 trials in a block. And within order memory is always tested for the second and the 6th item of a particular event. So this is an animal event transitioning to our apparel event. Okay, so we asked, did horse come before fox? This is our within order comparison. And that's either right or wrong, right? So a 1 or a 0. And we use a logistic regression decoding analysis to try to decode whether transitions between individual words affect whether participants will get this order memory question right or wrong. So you can see here, we do semantic similarity between dog and horse, or between the first and the second item, horse and bear, bear and cat. And then from the next event down, from our apparel event, from belt to fox, belt to bull, belt to cat, and so forth. And what you see here is that the middle items, how semantically similar they are within an event, drives order memory significantly through our decoding analysis. And in addition, how semantically dissimilar words on the boundary are, backwards drives temporal binding as well. Okay, so you can see that if I go semantic similarity from 6 to 1 or from 6 to 5, drives this order memory effect, but if I go backwards even farther, it doesn't. Okay, so let me conclude here. We first show that perceptual acoustic boundaries during a passive listening task in the auditory modality replicates event segmentation effects found in the visual modality. We go one step beyond this replication, and we show that self-reporting mnemonic strategies drive memory performance, but not segmentation. And finally, we show that semantic boundaries drive temporal binding effects, both within events and across event categorical transitions. Where we want to go from this is to do potentially an imaging study, looking at the different cues that are brought in by categorical and perceptual events to drive event segmentation, but also the common mechanisms between these 2 tasks. And we're working with Michael Sontenreich now to look at domain-specific features in music, such as cadences, and how those can create events in memory. Thank you, and I'd like to thank my co-authors, Michael Sontenreich, Keith Doling, Leila Davachi, and my advisor, David, for the contributions, and I'm happy to take any questions. Thank you.
Okay, cool. Thanks very much, Marie, for your great talk. That was wonderful. Let me check. I'll take questions. I did not see a question from Q&A for now. Can I ask a question? I missed the beginning part. Can you elaborate more about the temporal binding and the semantic boundary? Okay.
Sorry, I don't think I got the question there.
Don't worry. We can answer this question first. We got a question in the chat from Douglas. He said, "Great talk. What's the time interval between the participants being presented the sequence to be memorized and the recall testing?"
Yeah, it's quite slow. So like you hear a new word every like 3 seconds. The reason for that is because we wanted to replicate these visual studies. And so the presentation is quite slow and likely was strategically done for an fMRI experiment. I would like to see this done at a much faster pace. And I think in the auditorium, it'd be interesting to do this much faster, which is what we're actually trying to do now with music.
Sorry, can you hear me?
Yes.
Oh, yeah. So, so the reason why I ask, like, great talk, by the way, is that, like, do you think this is assessing, like, this effect of temporal binding in the time scale of short-term memory, or is it, like, a bit longer than that? Like, would you expect a change if you do the same test, like, a day after or 2 days or something?
Yeah, that's a great question. I tend to think this is still sort of a working memory task, short-term working memory task. It's not quite on the working memory side. Maybe if we presented it faster, it'd be like a fully working memory task. I don't think they'll remember the order much the next day without a lot more practice.
Okay, all right. Yeah, so it's
but now that we're speeding things up and we're doing a presentation potentially that's much faster in music, we're trying to lean more on the working memory literature, and that's what we actually think is happening.
Okay, so it would be like compressing the whole experiment to match that frame, the time frame.
Exactly.
All right. Very interesting.
Thanks, Douglas, for your great question, and thanks for the answer. We got the next question from Nico Regans. It's a great talk, Omri, great design, very compelling. I'm curious to what degree you think you can get the auditory boundary in fact with a single speaker, but with the background noise, ordinary context, changing across sets.
That's a great question. I think that you can get it because I think what you mean is, like, say there's like a cue at the boundary with a single speaker. Will you still see this? Now Layla's group, I forget exactly who was in the paper, it was a new Nature Communications paper that shows that if you have some sort of sound that's either in the right or the left ear at the boundaries, that also causes the same effect for visual stimuli. And I imagine something similar will happen here, but perhaps less robustly, because I think we have pretty strong intuitions about the switch of speakers. I think this creates quite distinct boundaries, but my intuition is that will also show similar effects.
Very cool. Thank you very much for the question. That's great. We have to move on now to have a second talk. Our next speaker is Douglas Fitosatomi. Douglas is a PhD candidate in the computational neuroscience and machine learning from Imperial College London. He's currently working on the memory formation and stabilization in the brain and continual learning in deep neural network. It's easy to find Douglas online too. Looking forward to your talk. Please go ahead, Douglas.
Hello?
Hi, we can hear you very well.
Oh, okay. All right, cool. Yeah, so thanks for the introduction. Yeah, so today I wanted to present our work on the dynamics of Ingram cells and systems consolidation. So the idea here is based on this experimental finding that Ingram cells in medial prefrontal cortex and in hippocampus, they behave differently. So in medial prefrontal cortex after training, Ingram cells in medial prefrontal cortex, they are in this silent state, meaning that they can't be recalled at a recent time point, but then over time, they become active and then now they can be recalled at a remote time point. And this process of going from silent to active is termed maturation. And then it is accompanied by this increase in dendritic spine density. But then for hippocampus engram cells they're encoded already in an active state, meaning that they can be recalled at a recent time point. But then over time, they become silent and they can no longer be recalled in a remote time point. So this process of going from active to silent is termed dematuration, and it is followed by this decrease in dendritic spine density. So our goal in this project is to investigate the mechanisms behind these engram cell dynamics. So we started with the simplest possible model, which only includes hippocampus and cortex. And then the idea here is to replicate engram cell dynamics in both of these regions, and also to capture the fact that the output of hippocampal engram cells is essential for the maturation that happens in cortex, as demonstrated in the experiments. So we start the simulation with a training phase where we present 4 distinct stimuli at random. Then we go to a consolidation phase where we don't present any stimulus, the network evolves spontaneously. And then finally, we go to a testing phase where we present partial cues. Then we measure the ability to recall the engram cells in both of the regions. So at this point, I wanted to point out that even though the synapses in the network are plastic, the synaptic plasticity rate is region specific. So meaning that the learning rate in hippocampus is higher than the learning rate in cortex. And also the synaptic consolidation time constant in hippocampus is also longer than the one in cortex. So here we can look at the degree of coupling between hippocampus and cortex at the end of the training phase. And then what we can see is that by looking at the mean weight strength between the engram cells that encode each of the stimuli between these 2 regions, that they have very strong coupling, such that after consolidation, when we do testing, we can see that here in blue, hippocampus has a very high recall immediately after training, and then has a very low recall after consolidation has taken place. So we have dematuration in hippocampus. But then now when we look at cortex, it already has a very high recall even right after training. Then after a small decrease, it goes back up. So this is not a profile of maturation as it should be, going from silent to active. And then here, if we block the output of hippocampal engram cells to cortex in the consolidation phase, we can see that we can recover a very high recall in cortex throughout the entire consolidation period. So this configuration here cannot display engram cell maturation in cortex as it should, based on the experimental data. So what we did was we fixed the connections from hippocampus to cortex. Now they're not plastic anymore. We go through the same simulation protocol, and then now, as expected, we do not have coupling between these 2 regions anymore, between hippocampus and cortex, since these synapses now are fixed. And then when we do the testing with the intact network, now we do have maturation of cortical engram cells, because initially here in orange, they're silent, and then later after consolidation, they're active. However, when we block the output of hippocampal engram cells to cortex, we still have the same profile of maturation, meaning that hippocampal engram cells are inconsequential for the dynamics in cortex. And this is contradictory to the experimental data. So in conclusion here, using the simple architecture with only hippocampus and cortex, we cannot capture this effect of hippocampus being critical for systems consolidation in cortex. So a solution to this is to include an intermediary, the medial dorsal thalamus specifically. And now we have this multisynaptic pathway that goes from hippocampus to thalamus to cortex. And then by using this expanded network, we can replicate dematuration in hippocampus, maturation in cortex, just like before. But then now the output of hippocampal engram cells is critical, is essential for the dynamics of engram cells in cortex, matching the experimental findings. So in this configuration here, we use these 4 set of stimuli, these 4 horizontal bars. And then after training, we go through consolidation, no stimulus. Then we go to testing, where we present only the middle 50%. And then in the consolidation phase with the intact network, here in orange for cortex, we can see that we have, again, maturation going from silent to active, and dematuration going from active to silent in hippocampus here in blue. And then we can see here that the model also predicts that engram cells in thalamus are going to be active at recent and remote time points. Now we can look at what happens to the engram cells during the consolidation period. So here in the last half an hour of the 24 hours of simulation in the consolidation phase, we can see here that in cortex, thalamus, and hippocampus, we have these coupled reactivations of engram cells. And then now we can do repeat the simulation, but then now block the output of hippocampal engram cells to thalamus. And then what we can see is that we no longer have proper maturation of cortical engrams. Instead, Here in our orange, you're going to have this small increase in recall in cortex, and then after that it quickly declines, and essentially you cannot sustain proper maturation of engram cells but by excluding the output of hippocampal engrams. So essentially what this is saying is that you need to have hippocampal engram cells active during consolidation for proper engram maturation. And then here, when we look at the output, the activity of the engram cells in the consolidation period, again in cortex, thalamus, and hippocampus, we can see that blocking the output of hippocampal engram cells disrupts that coupling that we had before in the control network. So the idea here is that the model suggests that engram cells in hippocampus are essential for dynamics of engram cells in cortex because it is necessary for coupling reactivations across these regions. So now we can repeat again our simulation with this network, but now we block the output of thalamic engram cells to cortex. And then we can see here that we're going to have a very similar effect, but in a slower fashion. So we have this increase in cortical recall followed by a decrease, but even though we have this initial increase, eventually, gradually it goes down. So essentially, we also cannot have, do not have in this case, a proper engram cell maturation when we block the output of thalamic engram cells. So these thalamic engram cells are also essential for maturation in cortex. And we can see that the way that they support maturation in cortex is also by coupling reactivations across the regions, because when we block the output of thalamic engram cells, we no longer have coupled reactivations between cortex and thalamus. And this is the reason why we have this decline in accuracy in cortex that we see here. So medial dorsal thalamus engram cells are essential for cortical engram dynamics, just like hippocampal ones. And now finally, what we wanted to study is the role of hippocampus in recent and remote recall. So what we found here is that the degree of synaptic coupling between medial dorsal thalamus and medial prefrontal cortex can predict the dynamics of engram cells in medial prefrontal cortex. And as a result, it can also tell what kind of retrograde amnesia pattern is induced by hippocampal ablation. So let's walk through this step by step. First, we have the training phase, just like before, and the consolidation. Now in the testing phase, we do it in two ways. One with the intact network, and then in another case, we remove hippocampus just in the testing phase to see the effect of that on recall. So at the end of training, we can measure the coupling from thalamus to cortex, and then we can see here that it is strong, relatively strong, and then after 24 hours of consolidation, it gets even stronger. So this gives us this profile of recall that we saw before, where here in orange for cortex, again, we have maturation going from silent to active cortical engram cells. However, when we remove hippocampus from the network in the testing phase, we can no longer recall the memory at a recent time point because recall in cortex is very low. But then you can recall the memory at a remote time point because by then we already have active engram cells in cortex. So this pattern is this pattern here configures a graded retrograde amnesia. But then we can control the degree of coupling from thalamus to cortex by controlling the synaptic plasticity rate between these regions. So by modulating this plasticity rate, we can then have very weak coupling from thalamus to cortex at the end of training, which does not change after 24 hours of consolidation. It is virtually non-existing, the coupling between thalamus and cortex in this case. And this leads to a very different profile of recall for cortical engram cells. You can see here that they have a low recall in recent and remote time points. So you do not have that maturation that we had before here. And then when we do the testing with hippocampal ablation, we cannot recall the memory neither in recent nor in remote time points. And therefore we have a flat retrograde amnesia pattern. Finally, we can modulate the coupling from thalamus to cortex to be stronger by controlling, again, the synaptic plasticity rate between them, such that at the end of training, you have very strong coupling, even stronger than we had before. And after 24 hours of consolidation, this coupling only gets stronger. So now we have a different profile of cortical engram cell recall here in orange. We can recall memories in cortex in recent and remote time points. And then now when we do hippocampal ablation, we can still do that, and therefore we don't have retrograde amnesia anymore. So basically we have an absent retrograde amnesia curve. So all this is to say that thalamocortical coupling, the degree of synaptic coupling between medial dorsal thalamus and medial prefrontal cortex, at the end of encoding is driving the dynamics of engram cells in cortex, and therefore the retrograde amnesia pattern that is induced by hippocampal lesion. And then finally, to conclude, the model was able to reproduce some key experimental findings in the literature of Ingram cells, specifically Ingram cell maturation in cortex and dematuration in hippocampus. The fact that hippocampal Ingram cells and coupled reactivations are necessary for proper systems consolidation. And we also make a number of testable predictions, and some of which here are Ingram cells in medial dorsal are active in recent and remote recall. The fact that thalamic Ingram cells are necessary for proper Ingram cell maturation in cortex. Fact that Ingram cells in hippocampus and thalamus are necessary for coupling reactivations across the hippocampus thalamus medial prefrontal cortex pathway. And finally, that thalamocortical coupling is predictive of the type of retrograde amnesia that is induced by hippocampal lesion. Thank you. If you have any questions.
You're muted. You should ask you.
Okay, I guess now I can. I thought it was my problem. I was not hearing anything. So.
I'm sorry, I'm muted. Thanks very much for your talk. That was excellent. I'm not taking questions from Q&A. We have a question from Shruti. Can you tell us a little more about the networks you have used and designated as hippocampus, cortex, and thalamus?
Sure. So if we look at this configuration here, which is the one that has all the three of them, you guys can still see my screen, right? By the way.
Yes.
Okay. Yeah. So so the way that we tell that a cell is an Ingram cell is that at the end of the training phase, we take the last 5 minutes of training, and then we calculate the average firing rate that is evoked by the stimuli. And then if this average firing rate over the last 5 minutes is above a certain threshold, in our case, 10 hertz, then we say that this cell encodes that specific stimulus. So any given cell can be an Ingram cell after a certain period of training. And then the same cell can encode multiple stimuli as well. And then the difference in between the network in hippocampus, thalamus, and cortex is purely in terms of synaptic plasticity rate and in terms of the afferent and afferent connections, as you can see here in the schematic. So in terms of what projects to what, and then and also in terms of the learning rate and the other parameters that control the synaptic plasticity rate in each region. That I hope that clarifies it a little bit more.
Cool. Thanks very much. Douglas, can you also click Q&A to see the questions? And Shruti also said one more thing more specifically, but I think you already answered that question. Yeah. So we go for next.
Yeah, I can see it here. The
question from Diogo Pat, yes.
Yeah, so have you considered to close the cortex thalamus CA1 loop via nucleus reunions? No, I have not. That's a very interesting idea. We were kind of like starting with the simplest possible model, and then we added things that that made the minimal circuit that could reproduce these Ingram cell dynamics. But certainly there's a lot of work to do, and then include other thalamic nuclei and also other cortical areas. That's something that we're considering for future work.
Okay, cool. Thanks very much. That was really great. But we have a very tight schedule, so now let's go for the next talk. Our last speaker is Dennis Levy. Dennis is a PhD student from Technical University Berlin. Currently, he's working on a mechanistic theory of systems of memory consolidation. Also, he developed the open source software called Brain2, CUDA. You can also find a lot more information about him online. Looking forward to your talk. Dennis, please take the stage.
Thank you very much. Hello, everybody. My name is Dennis, and today I'm going to talk about a circuit mechanism for systems memory consolidation that is based on two abundant features throughout the brain. And these are happy plasticity and parallel synaptic pathways. First of all, we know from all these amnesia studies in humans and animals that we need the hippocampus initially to acquire new memories, declarative memories. But later, for old memories, we don't seem to need the hippocampus for all kinds of memories. So there seems to be some kind of transfer from hippocampus to cortex. At the same time, this transfer seems to also lead to memory transformations. From something more episodic, detailed autobiographic memories, to something more general, semantic, structural knowledge. And this process is called systems memory consolidation. And while there are many phenomenological theories for systems consolidation, we don't really understand the mechanism. So today I'm asking the question what the mechanisms are for this memory reorganization and transformation. And I will introduce a theory that first of all assumes that this is not a two-stage process from hippocampus to cortex, but instead a multi-stage process. And I will start with explaining what the first consolidation step, or a single consolidation step in our model is. Let us consider simple forms of memories. That means cue response associations. And let's assume that such a cue response association is stored somewhere in our brain in a multisynaptic pathway, that is this blue pathway here. Such that after learning, if I present a cue, I have an input, a representation of the cue in the input area, which then leads to a representation of the response in the output area mediated by this blue pathway. Now, during consolidation, we assume that cues are reactivated in this input area, which will then reactivate the response in the output area. And with Hebbian plasticity in a parallel synaptic pathway, that is a red one here, now we can learn the same association, because we have a causal relationship between the cue in the input area and the response in the output area mediated by the multisynaptic pathway. So effectively, the multisynaptic path here acts as supervisor for learning in the shortcut. Okay, let me exemplify this little circuit diagram here in the hippocampal formation, where entorhinal cortex projects to CA1 through two pathways, one via the shuffle collaterals, which is a multisynaptic pathway here in blue, and one directly through the perforant path. Now let's take the simplest case. That is, we have one single neuron in the entorhinal cortex and one single neuron in the CA1. And our cue response association is just if the cell in the entorhinal cortex spikes, then the response should also be a spike in CA1. And we store this cue response association in the shuffle collaterals. That means before consolidation, the perforant path, if we have a spike in entorhinal cortex here, will not lead to a spike in CA1, just a small EPSB in the CA1 cell. On the other hand, the same spike reaches CA1 with a little delay because of more processing here through the shuffle collaterals, which is now already tuned, which has stored the memory to produce that postsynaptic spike in CA1. If I now have spike timing dependent plasticity in this perforant path, I will potentiate this one synapse because I have a before post situation, even though the post spike was created by the shuffle collateral input. Okay, now after potentiation and after consolidation, if I now have a spike in entorhinal cortex, the perforant path alone can now trigger a spike in CA1. All right, that's a very simple toy example. Let's make it a little bit more complicated by saying we have 1,000 neurons in entorhinal cortex and 1,000 synapses from entorhinal cortex through both of these pathways. And the only difference is, again, we have a little temporal synaptic transmission delay. So we don't consider any computation actually in CA3 right now, just for simplicity. Okay, and now we store a certain synaptic configuration in both of these pathways. One in the shuffle collaterals, which is the memory that we want to store, and another one in the perforant path synapses. And now if we run our model and with STDP in the perforant path, what happens is that the weight configuration from the shuffle collaterals will be copied over to the perforant path. And for this consolidation process, all we have to do is randomly reactivate these 1,000 neurons in entorhinal cortex, and the STDP does the job. Okay, so this is a very simple example with the same inputs and same representations. Of course, in reality, the representations are often different, and we do consider this in a navigational task with place cell and grid cell inputs, but I do not have time to dig into this. So I'm going to instead focus on some experimental evidence for our circuit mechanism. And for that, first of all, we need to add the next step of consolidation. So if we take a look again in our hippocampal formation, where the perforant path was just the first parallel shortcut, right? This perforant path is also a multisynaptic pathway that reaches subiculum via CA1, which has another parallel shortcut pathway here in green, which is the perforant path that reaches subiculum from entorhinal cortex. So we can reiterate the same mechanism now to consolidate a memory from the PPCA1 to the PP sub. Now, if this was actually the case, that memories were consolidated like this through the PPCA1, then we should need the PPCA1 only for a specific temporal window for memory retention, right? So this looks like a temporary memory buffer. And Remondes and Schumann tested exactly that. So what they did is they trained rats in a Morris water maze task, where the rat has to find a hidden platform in a water arena. And they then lesioned the perforin path, the PPCA1 synapse here, at different times in this experiment and tested memory performance. And our model now can explain this experimental data from almost more than a decade ago, almost 2 decades ago. But I will start explaining what our model would predict and then come to the experimental results. Okay, so the way the memory performance is evaluated is by looking at where the rat swims in this arena and then counting the time that the rat spends in any of these quadrants. And the T here is a target quadrant where the memory, the platform was during learning, and that's where the rat should spend most time if it knows the memory. So our model now, I can't go into detail again for time reasons, but we assume that, so what we store here is now object place associations. And we assume that the place code is a noisy version of the actual place, such that we can use Bayesian inference to infer a probability map of the location given the neural activity, which gives us these kind of spatial probability maps that is basically the probability of the platform to be in any location. And we can calculate these maps from either only input from the Scheffer collaterals, only from the PPCA1, or only PP sub. And we can see that initially when I store an object place association for this platform here at day 0, the Scheffer collaterals learn this association. And then over time, this memory fades out while it's consolidated in the PPCA1 synapses, and over time it fades out again, and slowly it's consolidated in PP sub synapses. And now what we can do is we can test memories at different times. And what Remondes and Schumann do is they test, first of all, short-term memory. In the control situation, we have this task. The rat has just trained on to learn where the platform is. And now there's the memory test. And here now these bars show the time that the rat spends in each of the quadrant for the experimental data. And for our model, it shows the probability to be in that quadrant, right? And so what we find is for short-term memory, just after learning, the rat spends more time in the target quadrant. That's a correct quadrant, right? And then we can do the same test after 28 days, and it's the same thing. So we have short-term memory and long-term memory. And now we can ask, can we learn memories without the perforin path, the PPCA1 synapse? So what Remondes and Schumann did is they lesioned the PPCA1 synapse before training. Then they trained, and then they tested short and long-term memory. So if we test short-term memory, that means just after training with not having a PPCA1 synapse or synapses, then we still see the same memory as in the control study. So that means it seems like we don't need to need PPCA1 during acquisition for short-term memory. And our model explains this by showing that the memory is stored in the Scheffer collaterals and we don't need the PPCA1 anyways. But how about long-term memory? So what Remondes and Schumann showed, and what also our model shows, is that we lose long-term memory if we don't have the PPCA1 synapses. That is, usually the memory needs to be consolidated through PPCA1 and into PP sub. But if we don't have PPCA1, we don't have the memory anymore here in Scheffer collaterals, and that's where the memory is gone. Okay, now we can ask, do we keep long-term memory when we lesion the PPCA1, right? After we have acquired memories. So what Remondes and Schumann did is they now lesion the PPCA1 21 days after training. Okay, so, and before the lesion, they do a final memory test. And then this final memory test, this is, nothing is lesioned. You see the normal memory performance for the target quadrant, and the same in our model here. And then we can also do another test at 30 days, so 9 days after the lesion, after enough time in between happened for consolidation. And we can still see that the rat spends more time in the target quadrant than in the other quadrants. And we can explain that because at the time of the lesion, the memory in our model was already consolidated in the PP sub. Okay, with that, we have explained a lesion study on PPCA1 lesions. Okay. So far, so good. So now we had 2 steps in our consolidation model. Now let's have a look at the full consolidation model. Of course, we can iterate it as much as we want. And if we take a single step and iterate it more often, we can create a full consolidation model from between 2 areas, for example, from hippocampus to cortex. And I'm not going to go into detail here again, but if we decrease learning rates exponentially here with distance from the hippocampus, what we get is a power law in the forgetting curves when we look at the union of all the memory readouts in these shortcuts. So this is something that's observed in memories in psychophysical studies in humans. And it's something that's theoretically shown also by Fusi and colleagues. Okay, so this is now all the memory transfer, but what we actually want to look at is memory transformation. And I'm running a little bit out of time. So let's have a look here. We want to be able to acquire semantic memories from episodic experiences. And we say episodic experiences are just collections of categorical variables. For example, you're in the park, it's a sunny day, and you're with Bob and his dog. And some of these variables are random and some are dependent. So there's statistics in the environment, and you want to be able to extract these statistics, for example, that Bob is always with the dog. And we want to learn such semantizations in this consolidation model. But first, we're going to go back again to the single step to understand how this works. In the single step, we store episodic memories in the multisynaptic pathway, and then we consolidate them in the shortcut. And now learning all the details of an episodic memory is the same thing, or sounds like overfitting on all the details of a single data point in terms of learning in a neural network. So we do not explicitly model here one-shot learning as we do somehow, but instead we assume that after we learned an episode, we have overfitted on that episode. So we overfit episodic memories here, and then we do consolidation. And this consolidation now depends on the learning rate here. And we said it's a gradual decrease in this cascade, but for simple case, now we assume that there's a very slow learning rate here. And there we assume to get semantic generalizations. And now we store our episodic memories such that we leave one variable out and reconstruct the entire episode. And the way this works is every day we store multiple memories in the multisynaptic pathway. And then during the night, we reactivate cues in the input area to have consolidation. And now we can test both pathways for episodic and semantic memories. The episodic memories, we can leave out one of the random aspects of a memory and ask to reconstruct it in one of the training episodes. That is, I'm asking you for a random detail in some of your experiences, right? Or I can ask for semantic memories, that is for some of the dependent parts. So did you learn that Bob is always with a dog? And I ask that not in a test episode, that's something that you didn't learn. That's like asking, imagine you're in the park with Bob. What kind of animal would he be with? So did you learn the rule-based information? And then we can look at both pathways for both of these memories. So we said we overfit on episodic detail in the beginning in the multisynaptic pathway. So the memory accuracy is perfect. And then over time, we store more memories. So the old memories are forgotten and we forget them. That's a forgetting curve here. On the other hand, since we overfit here, we can't generalize well. That's usually a trade-off, right? So this pathway can learn the details, but it doesn't learn the rule, the general one. On the other hand, the shortcut pathway here learns so slow that during one day, we don't learn anything. And since every day these aspects of the episodes are random, we will not learn anything over time. But on the other hand, the semantic generalization, we will learn over time. And that is because in each episode that we learn, Bob is with a dog, and the teaching signal during consolidation is always Bob is with a dog. And slowly over time, the shortcut pathway will now extract the statistics of that variable. Okay, so far so good. Now we had the case where this pathway overfits on all the episodic detail and can't generalize. In this case, we need replay of the cues in the input area during consolidation because this pathway only learned the specific experiences. But now consider a case where this pathway doesn't only overfit, but it also learned to generalize. If this pathway also generalizes, then we can use random reactivations here. Because if you have random reactivations and there's Bob in there sometimes, then the generalization will lead to a dog response and we have the correct teaching signal. So we only need to replay when the multisynaptic path can't generalize. And we don't need it. Random reactivations is sufficient if it can generalize. Okay, and now that was a single step. If you go to the full model again.
I think we have to wrap up.
Yes, it's the last slide.
Yeah.
In the full model now, we have, of course, different learning rates, and we have more or less generalization and episodic memories. So we have spatial gradients here. Okay, and to summarize the whole thing, I've showed you how we can copy synaptic weights. I've shown you some experimental evidence in our theory. I've showed you the full consolidation model, and I've shown you how we can semanticize episodic memories. Thank you very much. And that's it.
Great. Thanks very much, Davis, for your great talk. We have a question in the Q&A. it's quite long. Can you click Q&A to check it and answer this question?
Yes, I have to stop the sharing Q&A. All right, let me see.
Yeah. Hopefully the Zoom won't kick us out in 1 minute.
All right, so the last one, cool talk. I'm a bit unfamiliar with the exact timings. Okay. I am actually not exactly sure. So I believe that the transmission delays are something like 5, 10, 15 milliseconds, right? And the way I think of LTP and LTD windows is as long as the presynaptic spike comes before the postsynaptic spike, you know, they're different paradigms or tests, of course. I would have assumed that it's still potentiation. So you're saying of thinking of STDP windows where when you have a higher transmission delay, you get into LTD. Because, okay, I see what you mean. I thought it is biologically plausible. I didn't choose it actually because I didn't do that exact simulation. I can't really answer it. Yeah.
Okay, cool. Thanks. We are running out of time. Thanks very much for your question, Jeremy. Thanks very much again to all our speakers. I'm about applause for once for all. And thanks very much for being here, everybody. And also, I'd like to thank Froza. She was helping us as a backend host. And cool, I think we did a great job. Okay, enjoy the rest of the conference. Congrats, everyone.
Guys, and thanks to the hosts.
Thank you, everybody.
Thank you.
Thank you.
Thank you.
Nice to meet you.
Well, bye-bye.